Welcome back. In my previous two videos, I discussed how quickly bacteria can grow when provided with a friendly environment and enough nutrients. A single bacterium like E. coli can undergo growth every 20 minutes if the conditions are favorable for multiplication. This means that from one bacterium, two will appear, and then both will grow and reproduce, and resulting in 4 cells. The cycle repeats continuously, and if so, after 48 hours with a doubling time of 20 minutes, it would produce a progeny of 2.2 into 10 to the power 31 gram. That is roughly 4,000 times the weight of the Earth. If the cells were to continue at their optimal generation time for 36 hours, bacterial cells would cover the surface of the Earth. However, nature has its own mechanism to control everything present on this Earth. In nature, nutrients are limited and there is intense competition among different species that utilize the same resources. Some parts of the world are dry, or there is very limited available water to sustain constant growth and multiplication, and during the day the temperature is high and at night it drops. Therefore, the natural environment is not favorable for unlimited growth. In the laboratory, we can prepare an artificial medium and provide friendly conditions for bacterial growth. Yes, they will undoubtedly grow exponentially until the medium is completely depleted. The cell number will increase over time. When we plot the data, the curve will look like this. However, when we use a single batch of growth medium, meaning no extra medium will be added and no medium or cells will be taken out, this is called batch culture. The bacteria will behave in a totally different way. So let's examine how they behave and why. When we prepare an artificial medium and pour it into a petri plates, culture flask or test tubes, and if nothing is added or taken out, the system behaves like a closed system. Now we will inoculate a bacterial culture into the growth medium and seal the container in this way. Air can only pass. The bacterial number will not increase immediately. Not only that, you will observe that the number does not increase exponentially for a certain period of time. This phase is called the lag phase. What do they actually do in this phase? Are they really growing or not? The bacteria are not dividing in this phase. They are just preparing themselves to grow. They are synthesizing proteins and other biomolecules for metabolism. And due to the accumulation of new biomolecules, their cell size, weight also increase proportionally. They also start DNA replication. That means bacteria are metabolically in an active state. The duration of the lag phase is species specific and depends on several internal and external factors like age of the culture. If the bacteria are old, they will spend more time in lag phase than young culture. Young bacteria and old bacteria sound interesting, right? Here young means they are actively growing and old means they have passed that phase and now are not growing or growing very slowly. Let me explain. E. coli divides after every 20 minutes if all the conditions are favorable for growth. And the bacteria is called young if it maintains this cycle or close to that time. When the cells are not growing after every 20 minutes and they are taking much more time, for example 3 hours, it will be called old culture. Another important factor is the composition of the medium. If the bacteria are already isolated from the same medium composition, then they will take less time to adapt. But if the bacteria were isolated from a totally different medium and now are inoculated in a different one, then the duration of the lag phase will be higher. They will take more time to adapt into the new environment. The duration of the lag phase also depends on the type of bacterial species. Some bacteria generally grow very slowly, like Mycobacterium tuberculosis, and the duration of the lag phase for these bacteria is longer than the E. coli. The size and shape of the bacterium are also important parameter. If the cell size is small, the bacteria will grow faster because the nutrient diffuse more quickly inside the cytosol due to high surface is to volume ratio. After the lag phase is over, bacteria will enter into the log phase or exponential phase. Here the bacteria will double after a specific time interval, that is called the generation time, and there is a constant increment in the cell number. They grow in the same way that we have already discussed. The phase is called the log phase. The bacteria obey this equation in this phase. You can calculate its growth rate, generation time, and other kinetic parameters only in this phase. So the log phase has significant medical importance. You can predict the nature of the disease, for example, chronic or acute infection, if the organism under study is pathogenic, and how long it will take to produce disease symptoms, you can also calculate. When they will spread and how quickly can also be calculated from this stage. The metabolic activity is high in this phase, and due to that, bacteria are often smaller than their usual size. They hardly get enough time to reach an appropriate size. If you want to isolate any metabolic product like amino acids, vitamins, single cell proteins, etc., you have to study this phase only. In this phase, the production will be higher. This phase is also called tropophase in industrial microbiology. Now the question is, for how long will this phase stay? It depends on the species. If the bacteria grow fast, they will finish the log phase very quickly because all the medium will be depleted and there will be no medium to sustain more number of bacteria. Not only that, the duration for the log phase is also depends on the medium composition. If the medium is nutrient rich, it will provide more number of bacteria. And if the medium is nutrient poor, it will not able to sustain more number of bacterial cell. The population density depends on both how often the culture is agitated and the size of the vessel. It was observed that after a certain culture density, due to increased competition for nutrients, bacterial growth stop. So if you want to isolate primary metabolic products on a large scale, you have to design the medium accordingly and also provide the other essential criteria. After the log phase is over, the bacteria will enter into the stationary phase. Here again, no increment in population size is detected. Here again, the question appeared, why exponential growth stop? At this stage, the nutrients are depleted and the toxic metabolic waste products accumulated outside the cell, that negatively impacting the growth of the microbes. Few bacteria that are strong enough compared to other can divide, but while others that are too weak, they stop growing. Competition for the same nutrients is intense here, and some bacteria start producing and secreting active metabolites that are lethal for others. For example, a soil bacterium called actinomycetes starts producing antibiotics in this phase. These biomolecules are the final product of the secondary metabolism and gives an extra advantage in adverse condition. This phase is also called idiophase in industrial microbiology. In that case, actinomycetes will grow, but on the other hand, other microbes will die. As a result, there is no visible increase in cell population and the line looks flat. This phase is also very significant in terms of medical and industrial points of view. Many of the secondary metabolites secreted by the bacteria into the external medium are bioactive and may have some potential role in disease treatment. Some of them are used as food additives and food preservative. In this phase, in order to survive, bacteria start adopting different shapes and arrangements, and because of that, it becomes hard to identify them. For example, spore-forming bacteria start sporulating very rapidly. At last, the nutrients are exhausted and the concentration of the toxic byproducts increases significantly, and because of that, the bacteria are no longer able to survive. They start dividing exponentially as observed in this phase, but here the slope line is negative and the rate is much slower than this. This phase is called the death phase, and it is also very important. We can calculate kinetic parameters like death rate, minimum concentration of the toxic material that can initiate the death phase, and how long the population should be treated to kill all bacterial cells. These kinetic parameters are used extensively in food, pharmaceutical, and for the production of sanitizers. At the end, in some bacteria, generally a fraction, not all the bacteria in a population of the cell can survive even after the death phase. They are adapted so well that in the adverse conditions, they can multiply very slowly. They utilize the nutrients released from the dead cell, and after a few days, the situation again becomes unfriendly for some bacteria, and they start dying. Only a few progeny that are better adapted for survival can only grow, and this phase is very dynamic. This phase is called the prolonged decline phase, and when we plot this data, we will get a curve like this. This is called the batch growth curve, and nearly all bacteria follow this. So that's all for now. If you like this content, press like, share and subscribe. And finally, thanks for watching.